We dive deep into knowledge in our own adventurous, joyful, and passionate ways. Like fish among coral reefs, we observe, record, and seek new ways for life to thrive. Biology offers diverse career possibilities. Researcher, biobrainer, bioanalyst, lecturer, and environmental consultant. Without accreditation, we achieve, we contribute, and we help protect the environment. We can become anything we
Tes, tes. Ya, Assalamualaikum warahmatullahi wabarakatuh.
explore the sea of knowledge to better understand and care for life.
Good morning. Good morning, everyone. Good morning. Our tes. So yeah, today we have a special guest. We have two special guests. We have Mas Edo and also Katalin. The both of them will give talk. So today and tomorrow at the same time at 9 until 12. So yeah, I think you guys already know about the topics for the talk for today. And the time window will be from 9 until 12. However, it will be very flexible, depends on Edo and also Katalin. So before starting the talk, I will give you some short introduction about Edo and also Katalin. Edo is our alumni. He did bachelor and master study here in biology of ITS, and also now he is doing PhD in Wien University in Austria, Vienna. TU Wien, Uni Wien, University of Vienna, and also Katalin did his bachelor in Wien as well. Your bachelor, I mean, your bachelor degree. In Krems, okay. And he also did his master in Edinburgh, right? And he is now doing PhD as well as same as Edo, right? Yeah. He is now owning a startup, so yeah, it's very great. So I think we can directly start the talk. Okay, so I will just give the time to Edo directly. Okay. So any one of you, if you have any question, please don't hesitate to ask. You can also ask in Indonesia. I will try to translate. Also, Edo also can try to translate. And then the answer we can also try to translate to Indonesia if you would like to have the question. And also, sorry for the inconvenience, Katalin and Edo. We have some technical problem with the AC. Air conditioning is not so good. So let's see, tomorrow we will move to the other room, or we will just still do it in the same room here as well. I don't know. We will try to solve the problem. Okay. Ruangannya full. Is the monitor behind already on? Sudah on ya? Okay.
In a single drop of life, we observe how the world grows and moves. Life is dynamic,
Okay, cool. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Thank you so much for coming. And also thanks to Pak Arif and all the faculty members that already provided and give us time to have a talk here. So today's agenda is going to be a lecture and also some practicality, and we're going to play around with this bioreactor later. But for now, let's start with this theoretical framework about the economic principles of microbial growth, which consists of allocations, trade-off, and quantitative models. So this talk actually funded by Cluster of Excellence at WF Austria as part of the Cluster of Excellence Global Ambassador Program. And today, I'm here with me with my collaborators, Katalin Rusnak, and he's owning the startup, and he's the one that is soloing this project by himself, which is really cool. So we're gonna go back into that devices later and the development and everything. So it's gonna be cool. And for those of you who are interested into entrepreneurship and then somehow trying to incorporate life science and entrepreneur, you're gonna be super stoked with this. Cool. As Pak Arif mentioned, I also come from Indonesia, and I did my bachelor back in 2018 under Pak Arif supervisions, which microbiome profiling in crude oil polluted soil for my final project. And then for my master degree, I also did it in ITS with Ibu Eni, utilizations of soil microbe to enhance mechanical properties of geopolymer for sustainable construction materials. And I joined University of Vienna back in 2024 as part of the Cluster of Excellence program with the project focusing on microbial growth, biomass, and carbon use efficiency. And also my colleague from Repli Factory and also IMC CRAMS as a scientific project staff there also will have a talk about the development of bioreactor itself. Cool, let's start with the theoretical framework. Well, to truly understand how a cell functions, we can view through the lens of economic system. So let's think about how the cell has high efficiency factory, and then we'll go back that later. But has anybody here has a thought about economic principles? Prinsip economy? Ceteris paribus, something like that. So there's something like a trade-off and then opportunity cost and something like that. So we're going to talk about that, but in a context of cell biology. And then I'm going to use mainly a factory analogy. We're going to go back later in details. And then also there's some like economic principle and other biological disciplines like opportunity cost of metabolism and then some metabolic pathway allocation, something like that. Well, the factory analogy here is super useful because you can think of cells to have converting inputs into outputs under certain constraints. So in a way that you have some limited resources and you have to be efficient about that. So that works the same way as a factory because you don't have like infinite supply of money, right? So you don't have an infinite supply of spaces, or you have to think about, okay, which machinery to be located in a certain factory, and do I have to increase more how to increase more productions? So you cannot force like machinery to that only capable to, let's say, produce one car in a day, and then you cannot push that. If you want to increase the production, then you have to increase the number of the machinery that's needed. So that's how it works as well with the cell biology. Well, if we talk about cell biology, it involves so many economic processes, which include the metabolic pathways, and then cell manage an energy budget by producing spending the energy currency, which is ATP, and then they have to perform the constant resource allocations. That's the main point. So the allocation in here is a really important topic, but we'll go back in detail later. And then storage and saving. And also waste management, which is quite cool, like reusing the biomass that you produce as energy sources as well. And then there's going to be like trade and transport, and then the divisions of labor. Cool. Now, economic terms are only useful if they explain in the terms of cost, benefit, and constraint. So I have two different systems here, but somewhat similar. The first one is microbial growth, and the second one is construction company. So you can think of it in microbial growth, the constraints could be like diffusion capacity, and then cell sizes, and then the cost could be like enzymes, nutrients, and toxic metabolites. And then also, what is the benefit? It could be like biomass productions and grow faster, you name it. And that also quite analog with the construction company. So think of it like you have a constraint in terms of like working hours, like the contract that you have with the developer and then the customer. So you have to be really resourceful about that. And the cost itself could be like a personals. You have to think about materials that it's going to be used, and then the machines as well. And the benefit is also about revenue, getting like a benefit from the constructions that you build with the customer. And then now I want you to look at this map. So you can, there's like question is, why? Okay, cool. The question is, why is the large percent of protein
Kamu masih di kos gak, Bi? Kamu berarti Enggak.
Tolong angkat tangan. Ya, aku melihat beberapa yang resen. Oke, yang jawab sel B. Interesting. Oke, cool. Kira-kira kenapa? Ada yang mau jawab dengan jawaban tadi? Kenapa sel yang tumbuh lebih cepat, dia itu lebih besar atau lebih kecil? Ada yang mau coba kasih tebakan? Anybody want to have a guess on why the cell grow faster could be larger or smaller? Ya.
Let me answer the questions given. Sure. I'm Fatir. I'm from I'm the first year. I'm a biotechnology freshman here. I think that the cell B has a physically larger appearance because of they invest more on the size of the cell instead of using that to replicate. It's if I could say simply, the cell be choose. Saya pakai bahasa Inggris. Jadi sel B ini lebih memfokuskan ke quality, ke kualitas atau ke infrastruktur dibanding buat ekspansi kalau misalnya di bisnis. Iya, yang B, sel B. Karena sel B ini dia lebih fokus ke apa? Dia heavily invest ke selnya pasti dibandingkan buat replikasi atau pembelahan sel. Oke.
Ada jawaban lain? Ya, yang di belakang.
Permisi. Mungkin kalau menurut saya sendiri untuk yang lebih besar mungkin sel A. Soalnya I've once read that a cell has the maximum sizes. Nah, kalau misalkan luas permukaan itu ada ukuran maksimalnya sendiri. Jadi sebuah sel itu nggak bisa terlalu besar. Soalnya akan volume perbandingan volume dan luas permukaan akan menjadi tidak seimbang. Jadi mungkin untuk menurut saya sel A lebih besar secara biomasa mungkin. Seperti itu.
Terima kasih atas jawabannya. Hahaha, this is interesting. Okay, before I give you an answer, I will start with the very basic fundamental principles about growth. It's called growth law. So there's this in the 50s, this is the fundamental view on the relationship of growth rate and mass. So you can see in the x-axis here is like how fast the cell is growing. And then on the y-axis is the mass or the volume or the RNA to protein ratio, you name it. So there's this positive linear relationship, which means that the faster you're growing, the larger the cell size would be. So this is, it's already been proved in the 50s, and then there's this like a ribosomal growth law as well back in 2010 by Scott and Manalis, which is really cool principles. And then why is that? So the faster growth means the higher RNA to protein ratio, which means that you have more capability to produce more proteins. So that makes sense why the faster growing cells is bigger. Because imagine you have a factory. You have a car factory, let's say. In a day, you can produce like somewhere around 100 cars. And then it only, let's say the volume of the factory is this biology building. Now, if you think about getting more revenue, then you have to increase more productions of the cars, right? Let's say you want to increase it up to 250. Then what is the logical answer for that? Then of course you have to expand your factory, right? You need more machinery. That's why you grow bigger. If you want to have more revenue, then of course you need to be bigger. That's also has the same effect on cells, because you need more machinery, you have to produce more proteins, and etc., etc. That costs you more volume needed. Now, this is getting really interesting because specific carbon source really alter the fundamental physiology of the cells. So in here, there's like a respiration rate and growth rate relationship. When you're growing on sugar, this is like efficient, but when you grow on acetate, it gets more efficient and efficient. But the question is that, why is that? So because they have like more pathway to get into, then you have like more path you have to go through just to get the energy. So that's the other reasons. Now, why there's this trade-off? Why, if you want to get bigger, why you have to cost another things? Like this is what we call trade-off. You get other things, but you lose some other things. So the reason why this trade-off is possible, because there's this temperature differences, pH, osmolarity, and also diffusion limits. So it doesn't really matter if you have like a larger cells, but you'll be limited by diffusions. Diffusion capacity is like there's a limit that you can bypass. So enzyme plus substrate have to collide, then of course there's this diffusion limit. And no known enzyme above the diffusion limits, you cannot like bypass that limit. So there's this trade-off because of that. And then there's also macromolecular crowding, which is the concentrations of macromolecules, how fast they're going to be colliding. And then there's also some limit in the cellular processes as its translations. Now, why is that? Like, why does thing happen? Because proteome space is limited. So you see previous graph on acetate versus sugars, let's say like glucose in here. You see that because of this limited proteome, then you have to allocate like how many percents that is going to be infested for certain things. For example, in sugars here, you have like a very short pathway compared to acetate, like the productions and everything is quite short. Then those allocation is more into ribosomes to build cells. But when you grow on acetate, the vice versa, you need more pathway to get into. So you need more investment on metabolisms. That's why you have like a smaller cell compared to when you're growing on sugars. Cool, we're gonna go to case study number 1. This is really interesting. because exponential growth doesn't mean a steady state growth or balanced growth. So you typically, so I think you'll be already familiar with the microbiology laboratory practice back in the freshman year. You grow your the culture, and then you measure the OD, and then you plot the graph. And this is the typical batch culture graph you could get from this batch culture. There's a lag and log and stationary and also that. But the problem is with OD measurement, they only measure the total biomass. So if cell size changes, the OD can rise even while the cell physiology is shifting. So I'm gonna give you example here. What is the steady state growth? Like what is balanced growth? So typically people assume that when you are in exponential growth, you are in the balanced growth. But that is not the case. So what is the requirement to be filled to be considered as a balanced or steady state growth? So first, the cell counts and the population fluorescence, whatever population volume, RNA to protein ratio increase the same way. Like they have the same, excuse me, the same growth rate. They have the same rate. So if you plot it here, it's gonna be look like this. So they have the same slope with all of this. So the rate of changes of the number of cells, the volume, the mass, and also the number of molecules inside the cells change the same, like exactly the same. So that's when we call it steady state growth. So if we take a look back to the OD measurements, there are like two hypothetical culture with same OD, but could be different cell mass and size. So let's say a culture 1, OD 600 is 0.3, and there's a culture 2, OD 600 is 0.3 as well. They have the same OD, but the case could be like one culture is many small cells, but or fewer larger cells. So you cannot really see it when you collapse it in the population levels with the OD 600. But there's a cool measurements that you can use using a single cells buoyant mass using a cantilever. So this is SMR instrument. So instead of getting full biomass, total or average mass, you get a full biomass distribution. So you will get like each individual cell mass. So it's not like a total. So you get individually counted. So instead of getting an average, you get like distributions, like the range of cell size could be. Well, how does that work? Well, imagine that there's a cantilever and then you etch the channel into that, and then you can pass through the particles into that. And this cantilever is vibrating at a certain frequency. To give you better illustrations, this is the animations on how this instrument works. So imagine there's this cantilever that vibrate in a certain frequency, and then whenever you add more mass, which is like a particles, it could be anything, it will shift the vibrational frequency. So these changes in vibrational frequency represent the buoyant mass. So over time, not only one particle passed through it, there's like 500, like 5,000 cells per 5 minutes, which is quite fast. You get like more particles, you get more vibrational frequency, and you get multiple mass, single cell mass measurements, and you get the population profile. Then the question is how to reach the steady state in a batch culture experiment. So my colleague were trying to see like the window of a balanced growth in a batch culture using this instrument. So they start with the culture streak, and then they grow the overnight culture, and then they separate it into 3 different dilutions as a starter. So 100 dilution fold, and then 1,000, and then 10,000s. So this is the experiment, and then they will measure the OD, they measure the mass as well until the stationary phase. Okay, this look overwhelming, but I will get you through it. Let's see it by one. Excuse me. Cool. The x-axis here is the time, and then the y-axis is the buoyant mass in femtogram. So In each time point, you do the measurements. You do the single cell mass measurement, and you'll get the mass distributions. And over time, as the culture grow, they started to add more mass. Oh, all good. Terima kasih, Pak. And then you compare it. So there's like 10,000 dilutions, 1 to 1,000 dilution, and 1 to 100. So you see the differences in here, right, in terms of mass. And then I want you to look closely in this blue box over here. You see that it gets flattened. They becoming steady. But on the other hand, in the 1 to 1,000 dilutions, you only have like a small window there. They start to slowing down and then they get smaller. And then on the 1 to 100 dilution, you even get smaller window, like a shorter time period when you are in the steady state. So the distributions only stabilize after 2.5 up to 4 biomass doublings and only under strong dilutions conditions. And then if you take the median value instead of like a whole distributions, and you can plot it like this. And this is the getting larger and larger, and then up to the exponential phases, they get steady. So they have the exact same mass there, but only this small time window here. It's roughly 1 to 2 hours. So if you get like smaller dilutions, like larger dilutions, you have like even shorter window of a balanced growth. But even in 1 to 1,000 dilution, you don't even have like a balanced growth. You haven't reached a balanced growth because the nutrient is already depleted. Now, when you compare the OD is exactly the same, right, in here. You can see here that OD is roughly the same, but the mass is totally different. This is all the same culture. Why is that? Well, because when you start with a very low number of cells, then you have a time and you have more nutrients available until you reach the steady state. So if you start with a larger number of cells, you haven't reached the steady state yet, but the nutrient already depleted, and then you get smaller and smaller, and that's the problem. Now, why this steady state is important? Because you need it to be reproducible, right? And then the question is how to prolong the steady state window. So there's one way: you keep diluting them. Like after a few times, you have to dilute them, and then you can reach the steady state. But that'll be a problem. But another alternative could be bioreactor. You can use a chemostat where you supply the nutrient every certain time points, and then you take out the waste. So then you can reach the steady state, the balanced growth. Now, why it's important? Because if you have like a new culture and you want to have the profile of the metabolic capacity or metabolic production of a certain metabolite, then you need it to be in a steady state growth because you need it to be accurate, and then you want it to be reproducible as well because gene expression, protein synthesis, and cell composition are going to remain stable. Excuse me. And then growth law and physiological models assume that you take the data while in steady state conditions. And then, for example, if you want to grow like more practical, like in industry, you need like industrial efficiency level for metabolite productions. Then you need in steady state growth phase. This correlated? Yeah. So.
Hi everyone. Can you hear me? Yeah. I'll send you a link, but without the video. I mean to open Zoom, okay? think it's a. I might. Mhm. Yeah, I got my hotspot. I cannot. Okay, I think it works. Yeah, yeah. Do this. Does it work? Mhm.
Send this Google Doc thing.
See the. Okay. So, hi everyone. Thanks for the patience. Mhm. Today. So, I think I'll let Edo try to fix the screen while I introduce myself. I'm Catalin, and it's my first time in Indonesia. I really like it so far. I think it's a beautiful country. I'm really, really happy that Edo invited me to join him on this trip. And as you'll see a little bit later in the slides, I was born in a small country called Moldova. Did anybody hear about Moldova? One person? No? What I can tell you about Moldova, it's a small country between Ukraine and Romania. And it's very small, but it has very good soil. And as such, it was a major producer of wine in the Soviet Union. For its size, it was exporting a very, very large amount of wine. And we have the biggest wine cellars in the world. So they're actually drive-through, it's like a McDonald's drive-through, except it's a wine cellar with big barrels. That's one of the most exciting things about my country. After that, I decided to move out to Austria, where I did my bachelor's in biotechnology in a university not very far from Vienna, about 1 hour away. And after that, I moved to the UK, to Edinburgh, to study bioinformatics, because I felt like I needed a bit more math and programming. And combining biotechnology with programming is, of course, naturally bioinformatics. And then it so happened that I moved back to Austria to start a PhD, which I did not finish in the end. But I'm very happy with the progress that I made while I was working on the PhD. And specifically, I built this device that also Edo is now using. If it's not working, maybe we can. Maybe I can send you the except not the video. It's, uh-huh. Okay. Okay. Mhm. So can you tell me a little bit about your level of English? Who feels like they're above 90% English level? No one? Huh? I've heard at least a few people speaking English, and I'm sure you're above 90%. Like, you
Ada yang pede,
who think?
bahasa Inggrisnya 90%. 90% English you can talk, or you can at least understand what he's saying.
Okay,
Yeah.
okay. What about 80%?
80%.
Above 80%?
I think we are very humble in this case.
I
but
also think so, yeah.
is it working now?
Okay. Good. Perfect. So, yeah, this is where I'm from. So you can barely see, this is a map of Europe. Moldova is here, Austria is here, United Kingdom is here, Edinburgh. And in Edinburgh, I studied how to analyze mutation data in genomes. And in Austria, I worked on biotechnology and evolutionary biology. So what's the motivation behind this device here that Edo mentioned? You can play with it later. Now I'll show you a little bit. So I wanted to study evolution, how bacteria adapt to stress. And to do this, you take a bacterium, you hit it hard with something that stresses it, usually something like an antibiotic. And when you do that, it adapts, right? It fixes some mutations in the population that make it resistant to this stress. And then you can use bioinformatics and DNA sequencing to look what mutations made them resistant to this stress, right? And I was looking not at antibiotics, but at metals. So I was asking, how do bacteria become resistant to heavy metals like cobalt and nickel? And one beautiful result that we found is a mutation in this channel that lets cobalt ions through. So if you stress a cell with too much cobalt, which is toxic in high concentrations, it makes sense that it will knock out a protein whose only function is to let cobalt ions through, right? It's like a pore that lets something toxic inside the cell. You knock it out, you have less of this toxic atom inside the cells. And you can see it because we found a stop codon inserted at this location. Right, once you insert the stop codon, the protein translation stops here, and almost the entire part of the protein does not get transcribed. Yeah, so this is one beautiful result. You can read in more detail about it on replifactory.com/mutations. There's a more detailed analysis if you're curious into that kind of stuff. So how can we actually achieve this in the lab, that you adapt the bacteria? You let them grow, and then you hit them hard, which is illustrated here in red. And when you do that, you see the optical density is dropping when you dilute the culture, because it is not growing anymore. But at one point, it does start to grow. And when it grows fast, this is like a signal that you can increase the stress because they've adapted a little bit. When you increase the stress, they stop growing again. And after a while, they start growing, you can increase the stress further, and so on, right? So illustrated, it looks like this. You take the bacteria with something, you hit the bacteria in the head with something, it makes a small shield, you hit it harder, makes a better shield, you hit it harder, and so on. And you can do this by hand with a lot of pipetting and a lot of glassware that then needs to be washed. And after all that hard work in the lab, of course, you need to drink a lot of coffee. All right? But luckily, we have automated coffee machines, automated dishwashers, and you see where this is going, right? We build an automated liquid handler that does the pipetting instead of us. And a diagram is like this. We have 7 growth chambers, which are independent. Each of them has a magnetic stir, which mixes the liquids with an optical density sensor. So there's a laser that shines through the culture, and depending on the attenuation of the laser, we can measure how many bacteria there are. And we can control the media composition here using these 3 pumps. One pump adds fresh media. One pump adds media together with a drug, like a cobalt or antibiotic. And the third pump removes the excess liquid into waste. So you can make dilutions in each individual vial, and you control where the pumps direct the fluid by these gray valves. So you close 6 of them and leave just 1 open, and that's how you know that everything that the pumps are pumping goes to 1 vial, okay? It looks like this, and it's made using 3D printing. Who knows about 3D printing? Nobody? Who is listening? Okay, at least one person listening. Anybody familiar with 3D printing? I'm sure there's more people who are familiar with 3D printing.
Ada yang tahu tentang 3D printing? Just knowing is fine, I think.
Yeah,
By the
you
internet.
don't have to have a 3D printer. Just like having seen something on the internet is enough.
Yeah.
You're too humble, guys. And the 3D printing is nice because it allows you to build a very efficient body, and at a very low cost, right? So with a cheap instrument, with a cheap instrument, you can design and print your own parts that do a function that you want very, very quickly. And inside the device, there is a Raspberry Pi computer. Who has heard of a Raspberry Pi? No one? Okay, this is not as well known as 3D printing, but it's a very low cost computer that you can buy for about $50. That is really a fully functional computer. It has Wi-Fi, it has Ethernet ports, it has a CPU, it has USB ports. You can connect a monitor to it, you can connect a keyboard, and it's used inside the device to run all of the like heavy calculations and to serve a graphical user interface. So this computer is what handles all of the computation. Next to it is a custom designed board that controls all of the motors inside the device. How many motors do you think are in the device? Who thinks more than 5? Who thinks less than 5? Come on, guys, it's a binary. It's the sum of raised hands should be the total of this room. Who thinks it's more than 10 motors? No one? Okay, it's 17 motors inside. It's 7 valves, 7 stirrers, and 3 pumps. And there's actually one more. It's the cooling fan, because the Raspberry has active cooling here. The fluidic side is what you need to sterilize. Can anybody tell me why we need sterilization in biological experiments? Now the microphone goes to you. Who can answer this? Why we need sterilization in this kind of experiments?
Kenapa butuh sterilisasi pada eksperimen ini? You can also answer in Indonesian and then I will try to translate for you. Ada yang mau jawab pakai bahasa Indonesia juga boleh. Malu-malu guys. Don't be shy.
It's okay. I promise I don't bite.
Kenapa di eksperimen ini kita butuh sterilisasi? Why do we need sterilization in this kind of experiment?
Have you heard of contamination? What does sterilization do? Yeah, do you want to?
The real purpose of sterilization is to reduce contaminants in the sample. So we could track the right sample instead of the contaminants.
Sterilization is necessary so that we know that when we make a measurement with the fancy instruments that Edo has access to, you want to be sure that it's actually that bacterium that you're studying. Our bodies are full of bacteria, like head to toe. We have actually more bacterial cells in us than human cells because they're so small and so many. So it's really, really important that when you're running experiments, you're analyzing not what grows in your hair and on your skin, but what you took from the freezer in the lab in a very controlled way, right? And can you give me some methods of sterilization?
Ada yang bisa menyebutkan metode
How can you
sterilisasi?
sterilize stuff in the lab? Yeah.
Just raise your hand. Metode sterilisasi? Bahasa Indonesia.
So maybe there's method of sterilization that I do know. First of all, is that using alcohol or some kind of
Yes.
cleaning substance
Yes.
to remove microbial. The other one is to heat, using heat. For
Yes.
example, like
Yes.
in autoclave.
Yeah.
I think there's some kind of filtration method that can be used also, but I don't really know that much.
Yeah, so you said two. One is with ethanol because that kills the cells. One is with heat. And I can tell you a few more. One is with the radiation. Right, so if you buy plastic, who used plastic single-use stuff in the lab? Like, do you have labs? Yeah, and you use single-use pipettes, right? So those are typically gamma ray sterilized. So inside the packaging, they go in like a gamma ray sterilization machine, and the gamma ray just kills everything that can survive. Okay. Very good. Yeah. These methods are necessary to make sure that everything is sterile and only our bacteria of interest are growing. And in the case of Replifactory, the very nice thing about it is that the fluidics is easy to autoclave. So you can remove it as a closed system, just put it in the autoclave where it's heated up to 121 degrees, and the steam at this temperature and high pressure kills off every living organism. And the whole device was designed for exactly this procedure, that it has really a lot of silicone tubing that I'll take out from my bag at the end, and you can touch it, you can see it, but it's very easy to attach and detach from the instrument. So if something happens during startup, if you put your finger where you contaminate, or if you open the vial and something gets contaminated, it's not a problem. You can very easily autoclave, then you're sure it's sterile. Okay. Now I will show you a demo of the graphical user interface. It's 2 minutes and it's very fast paced. So if you look at the monitors, you'll get an idea of how it works. So we have this buttons. I think I might need to, wait a second, I might need to. Yeah, I'm gonna stop this. Mhm. Uh, Sorry. Okay. I think I might need to restart it again. Mm. Uh-huh. This was. Mhm. Yes, that was the problem.
Ayo,
I did
mahasiswanya
restart.
yang aktif ya.
Mhm.
Nggak usah takut
Okay,
salah.
hopefully it should work. Yes, okay, good. Sorry for the technical hiccup. So hopefully you should all be seeing the, I think now you can, yeah. Huh? Yeah, is it? Okay. I think now I'm also sharing the screen if you, so, ready? The user interface is like this with the 3 pumps, the valves, stirrs, and optical density. When you open the button, when you press a button to open the valve, the silicon tube opens, and then you can run a pump that adds the liquid over here. And the magnetic stir works like that. You can adjust its speed. And the optical density works with the red laser that you will see here in a second. And you can see that here the liquid is clear, the laser is very visible, and here it's not so visible because there are bacteria growing in these vials, right? And there's also a simulation environment where a mathematical model is used to predict approximately what is going to happen in the experiment and how bacteria will grow. And that lets you estimate how much media you need in the bottles. Because if you dilute them very frequently, you might need 100 liters of media in one day. And that's very difficult to prepare. So you need to adjust the parameters of the device so that the experiment is manageable. And that you can do very easily with a simulation environment. And then you say, we have these amounts of liquids in the bottles, press a magic button, and the experiment starts. And when the experiment is running, this is what's happening. You see the pumps. Every so often they spin and they add a little bit of liquid in one of the vials, right? They can do it at a very high frequency. In the experiments that Edo was doing, he was doing at a frequency with a period between 7 and 60 minutes, something like this, right? So yeah, so if you imagine making a dilution every 6 minutes by hand for many hours on end, it's not the nicest thing to do, right? So so this is what can be automated with this kind of devices like a chemostat. Yeah. Should I maybe give you back the opportunity? And yeah, let's keep the development part for then. I think it was, yeah. So later, maybe tomorrow, not sure when, I can show you a little bit also about how I developed this device to the state it is now. So it didn't start like this. It was a much less developed device. And I've gone through many cycles of iteration, and that's something that I can also talk to you about. But now I'll give the microphone back to Edo, where he can continue telling you about his experiments and how this chemostat helps him do great science.
Cool, thank you, Katalin. Back to the theoretical framework again. Right. Katalin already showed you like how to keep up with this experiment and then how to automate it. And that's very cool devices, and I love it because typically with the large bioreactor, I only have like 1 data points per week. But with Katalin devices, I can get up to like 14 data points per week, which is like way faster than I can expect. Imagine like how efficient is that. Thank you, Katalin, for developing this. This is really cool. Cool, now we're moving to case study number 2. We already have a view about the growth law, but the question is like, how universal is the growth law? Like, can it apply to like many different systems? Can it also apply to nature? And can you somehow use that to predict like how fast bacteria in nature growing, like in situ? Well, I'll start with a reminder about the growth law. So there's this relationship of the growth rate and mass. The faster you're growing, the higher the mass, the higher the volume, the higher the RNA to protein ratio, etc., etc. Now, this is a fundamental basis of that. But we don't really know like what if they're limited with something, because organismal growth is limited by elemental balance as well, like by nitrogen. Oh, okay, that's interesting case. This, no? That's interesting. Okay, let's go back a little bit. Okay, there's this growth law. There's this positive linear relationship of growth rate and mass. The faster you're growing, the larger you get, the heavier you get, and also you have more capacity to produce protein. Now, the problem is in nature, organismal growth is limited by elemental balance as well, not only by energy. So you can be limited by nitrogen, you can be limited by carbon, and also phosphorus. Then the question is like how growth law applies to these situations. Because in batch culture, you're typically limited by energy. You're limited by the type of energy you're growing, like the quality of the carbon source. So you have everything you need, but you're only limited by the thermodynamics capacity of the certain nutrients. But we're going to be talking about the nitrogen, carbon, and phosphorus limitation as well, how it affects the growth rate. The growth law, sorry. Because microbes in nature often limited by N or P. And then the question is like, how universal is that? And then this chapter asks how growth law change when N and P becomes limiting, not only by C or energy limited. Okay, I would like to remind you about the inputs of carbon, nitrogen, and phosphorus inside the cell. So carbon makes carbon precursors, nitrogen as well, to make amino acids and making proteins. While phosphorus come into this nucleotide, different inputs, and then you're getting RNA or DNA. So it's totally different bottleneck you will get depending on which elemental balance that limited. Cool. There's this very cool study by Chung and Lee, 2018. They're modulating the E. coli growing in chemostat in different growth rates. So they varying how fast the E. coli growing and also different conditions, C limitations and limitation and phosphorus limitations. Now it gets really interesting because when you are unlimited, you have more ribosomes and but you have slower elongations. But when you're C limited, you're somewhat similar because you have like the same input, right? So the CNN limitation is somewhat similar, but you have like extra inactive 70 as reserve, that is going to be used for a fast upshift later. But the interesting thing is that when you're pre-limited, you have fewer ribosome. Why is that? Yeah, because it's super expensive. RNA and DNA is super expensive to make because you're limited by phosphorus. But interesting thing is that mostly active and efficient. They're super efficient and active. Cool. The way I approach this problem is that I use a free-living bacteria in bioreactor and then measure the mass, growth, and metabolisms. Okay, to control how fast bacteria are growing, there are two ways. The first one is using the dilution rate in chemostat, and the second one by limiting them with C, N, and P limitations. So you're modulating how fast they're growing by controlling their in glucose, designing some other medium with different carbon sources as well. And then you'll measure the respiration rate and then varying the dilution rate, which is varying the how fast we're growing. And then you will do the cell count, you'll do the single cell mass measurement. So each individual cells will be weighed. Jadi bakalan ditimbang tuh satu-satu sel ditimbang. And then what could be expected from that is that the higher the dilution rate, the higher you're growing, the higher the cell mass would be. And then that's also applied to metabolic rate as well. And also depending on the carbon sources, you'll have like different efficiency as well. Cool. Now this is batch culture. What you see in here is the Vibrio data. So in our lab, we have like 6,000 Vibrio culture, many different species. And then we're trying to grow them in many different media and many different carbon sources to modulate the differences of how growing. So the x-axis is here, how fast you're growing. And then the y-axis is how weight, like the mass, the volume as well. So in here, you see the nice relationship in here. It means that the growth law also applies in Vibrio. The faster you're growing, you also have the higher mass. Now, they also have a positive relationship. Then the question is like, how does it look like when you're limited them with C, N, and P limitations in chemostat and modulate them in chemostat in bioreactor? So this is my data here that is overlap in the graph. Now, I want to guide you with this imaginary line here in the 0.4 x-axis, which means that in here, this is the growth rate, 0.4 per hour. Now, let's take a look into many different points here. When you are P-limited, you have a larger cell. And when you're N-limited, you also have larger cells. But when you are unconstrained, not limited by any elements, only limited by energy type, you have like a smaller one. So this is in batch, the gray one. But when you're C-limited in chemostat, you have way smaller mass. And why is that? And I think this is really interesting because I also have no answer why that thing happened. I haven't do any proteome analysis yet. But my assumption is that the growth law are insufficient. So you need more general view on how this thing can happen in nature. Now, we already established a clear relationship in ideal lab monocultures. And the question is that because in ITS, you're more likely trained to be an engineer, you'll be asked like, okay, what's the practical application about it? Like, how is it translated to the environment? So because you already have like the relationship of growth versus mass, you have the CNP stoichiometry, and also metabolic rate versus mass. Now you want to know like what's the overlap, and can you use that to somehow predict how fast you're growing in nature? So because right now, how would you imagine like taking a bacteria in nature, like how fast you're growing in situ? You have like what kind of way of doing that? Because they are in community level. You cannot like easily separate them and then see in nature how fast you're growing. So with this, we can predict that. Now, let's start with another basis of plankton paradox. I think you might hear about it in Ecology 1 or Ecology 2. So this paradox shows that in nature, with the limited resources, the expectation is that, okay, you will have like a less diversity. But turns out the empirical observations show another view, which is like more diverse. Like, how could you like have more diversity when the resources is very limited? And that could be about like resource limitations or niche partitioning, or even environmental fluctuation and filtering. Now that might happen in nature. Now, I want to show you like why mass is very important in microbial ecology, because right now in microbial ecology, when we talk about microbiome research, typically people only see the relative abundance of DNA from environmental DNA, not even seeing the microbial physiology. And I want to show you why it's important. In marine environment, you have a typical SAR11 clade and also Fibronacea. Typically, SAR11 is the most abundant one. They can up to be 10 to the 5th per milliliters, while Fibronacea only 1,000 per mils. They're like significantly less than SAR11. However, if you see their mass, it's totally different. Fibrio here is way bigger. It can be like 100 times bigger than, 10 times bigger than the SAR11, which is really interesting. Like, why is that? How could you like be larger, but in terms of number, you have less? Then that could mean when you have like a larger mass, you grow faster. Then the turnover in the ecosystem is also faster, and then the contributions to the carbon cycle is also higher than the SAR11. So this is why this thing is important when we considering the ecosystem functions. Now, we want to know about the community level physiology that may be predictable from monoculture-based rules that we see in the lab. So instead of asking like, okay, we have the growth law for monoculture, but does it also apply to community level? Well, we don't know. But my approach is that let's take like inoculum starter, like a natural community, and then grow them in chemostat and set them in the varying different rate, and then do some sampling later, like the first chapter that I shown you before. Now we can use fluorescent sorting for the population. So imagine you have like a mixed community, and then you can label them, and then you can like sort them. And then after that, you can do the downstream analysis, which is the 16S sequencing, just to make sure that you get what you want. For example, like Fibronacea from the rest of the bacterial community. And you can also do mass measurement of sorted cells. So with my colleague, Ben Roller, we're gonna be working on that. Now, what's next? You have like all this relationship about the growth rate and mass. The question is like, can we use these informations to predict how fast you're growing just by only knowing the mass? So there's also this our study that already available on the internet on bioRxiv. We're trying to grow a natural, the wild type of Vibrio in natural seawater and see how fast they're growing in their natural water. So as we can see here, there's this overlap of the wild Fibronacea, which is the red box here, and then the Vibrio isolates in seawater. And this is the Vibrio isolate that is starved. So this overlap here shows that they have the same mass. So there's this possibility that we can do like a reverse engineering by only knowing how much their weigh, like how their mass. We can see like, okay, approximately they're growing this fast. So that's what we're gonna do. And then the next, we are going to incorporate the C, N, and also P ratio as well to have a more accurate predictions on how fast they're growing in nature. So that's what we're gonna do. I think that's it from us. I would like to thank my supervisors and my collaborators, GMF, especially FWF, to give me funding for this. And Domis and Lambda, and also Replifactory, which with a really fancy and cool bioreactor. And I'll give it back to Pak Arif.
Yeah, thank you very much. Please give applause for Mas Edo and Kathleen. Thank you for the very exciting talk, both for Edo and Kathleen. If you have any question, please raise your hand. You can also ask in Indonesian, also in English. Doesn't really matter. Any question? Yeah, I think before the students, just for me as a bait, I have one question for both of you. So for Kathleen, I'm wondering whether your bioreactor, if by chance or by accident we spill some culture in, not inside the bottle, but outside of the, is it really autoclavable or how do you sterilize it? Ah, that one is sterilizable, okay. That's fine, that's fine. Okay, yeah. Uh-huh. Ah, okay, okay. Mm-hmm. Okay, okay. Ah, okay, okay. Ah, very nice to know. Yeah, because some people have very clumsy hand like me, so there is a high chance that we spill on the machine, which seems very expensive because it looks so sophisticated. Ah, okay. Okay, okay, yeah. Okay, okay.
And that is definitely way cheaper than the other bioreactor. For example, only for the vessels that cost around 3,000 euros, only for the vessels, not including the machine and everything. This one is like considered as super cheap with this fancy like seven chambers of working.
So it has all the sensors that you need to measure everything. Ah, okay, okay. So yeah, in case anyone is interested to to perform any sophisticated experiment like what Edo is doing, you can try to use this. Yeah, thank you for the answer. And another question for Edo, because of I'm interested with the machine that you show for the measuring the single cell mass. I'm wondering for the fast growing cells compared to the low growing cells, whether it has something to do with the percentage of the population where the fast growing cell has, for example, when you take the samples at the same time, the fast growing cell has maybe around 60 or 70% cells are ready to divide. So it's almost the septum is ready to finish, while the low growing cells have very laid back or slow growing, or the most of the cells in the population are still starting to wake up, for example. Is it related to that or just the number of the cells or the size of the cells in general?
I'm gonna show you some data from that I have here. Oh, so second. Cool. I'm gonna share some of the data
Take
that
your
I
time.
have.
Ya. Tanya pakai bahasa Indonesia.
Cool.
Mantap,
This is
ya.
one of the example. So the lowest part here is like the slow growing one. It grows like 0.1 per hour. And then this is like 0.2, 0.3, up to 0.6 per hour. And this is controlled. It doesn't really matter. So you see different distributions right there. Like in here, it's the median is like 61 femtogram, 53, and increasing up to 86 femtogram. And related to Pak Arif questions, like the cells might have different phase, like they're about to divide, they're just like after dividing. So that gives these distributions. So sometimes you'll have very unique distributions where you have like, instead of like a normal distributions, oh, no, should be this one.
Yeah.
Okay, this is the N, and this is the mass. So let's say this is like 0, 10 femtogram, 20, up to 100, let's say. So instead of having like one nice normal distributions, you would expect like having a binomial distributions, where another case would be like this and this. So you will have like two peak. You can imagine like the larger part, let's say this is 40 and this is like 20. Then you can imagine, okay, this is like a single cell, and this is the cell that about to divide. So you can have like a multiple different populations profile. So depending on the experiment, but most of the time you can separate distributions with a mixture model. So you take a log transfer them, and then you apply the normal distributions, and you'll get a very clean single cell mass distributions. So that's, I think that's not going to be a problem.
Okay, so it's already overcome with that bias, right?
Yeah, exactly. But another thing is that with this profile, you can also see like the range of cells could be, like the size of the cells could be. But the problem that I face, it's rather challenging. Say, I've been doing the statistics for like I don't know, for like months, and then it's hard for me to separate another distributions where you have like very long tail. That means you have like multiple cells attached to one another, or you have like multiple cells, and then you don't really know where to cut. Yeah, either in here or here or here, or it's kind of hard to really handle this kind of data. But anyway, you can also do the microscopy and then take like different volume distributions. You also have a volume distributions, and then you apply this to mass, because the proportions volume is proportional to the mass. Then I think statistically it's possible to take distributions, apply to distributions, and you'll have like a very nice mass distributions.
Okay. Thank. Is there any question from the audience? Yeah, finally, guys. Siapa tadi angkat tangan? Yang belakang dulu ya.
Okay. Excuse me, Miss. In the explanation of the GMO error, the machine was explained to be used for automatic dilution. So, what model is used? How does mathematics predict the result? Bahasa Indonesia. Jadi pada penjelasan tadi itu kan yang di game tadi itu dijelasin bagian mesin itu kan biar bisa pengencernya itu terotomatik gitu. Nah terus pakai modul itu apa ya? Terus habis itu untuk prediksi hasilnya itu bagaimana gitu? Terima kasih. Iya, katanya.
So the question is, in Kathleen's bioreactor, there is a dilution module. So this dilution module, what kind of module that you use for this kind of dilution things? And the way I understand the question is,
the way I understand the question is it the simulation model that predicts the growth of bacteria that I showed, or
Yeah,
the
the module that you use to make sure the dilution that you
The
put
pumps?
in, yeah,
the pumps?
yeah, the
Okay. So these are peristaltic pumps. And then can you show maybe, Edo? Yeah, if you hold it and if you tilt it like this. So there is inside there are 6 rollers that spin, and when this tubing is inside, it presses the liquid along the tube. So this is similar to how our intestines work. Or if you milk a cow, you do the same motion. It's called a peristaltic motion.
okay. To control the dilution or to control the flow, the peristaltic or the programming.
Yeah, so this is just a pump, and when it spins, it moves liquid through the tubing.
Okay.
And the valves here decide which of the vials the liquid will move to.
Okay, okay. I hope it's clear for you.
Maybe you can say this in Indonesian so that it's
Ya, oke. So, jadi di bagian mesinnya itu ada semacam rotornya yang bisa neken gitu ya, yang bisa menekan salurannya. Jadi gerakannya seperti aliran gerakan peristaltik untuk yang di usus atau di tenggorokan dan sebagainya, sehingga aliran dari larutannya itu bisa terkontrol. Clear? Sudah jelas? Oh. Okay, yeah. So every single tubes or flask is independently controlled, or
yes, yes,
Okay.
yes.
Okay.
Every time a dilution is made, 6 valves are closed, fully closed,
Okay.
and only one is open. So
Okay.
you cannot do a mixed where you pump in 3 vials at the same time,
Ah,
because
okay.
then you cannot know if they are all exactly split in 3, or maybe one of them gets a little bit more. But if you close all of them except one, then
Okay.
you know for sure it went in one direction.
Oke, oke. Thank you. Pertanyaan selanjutnya, siapa tadi? Ada yang angkat tangan? Kamu jadi angkat tangan enggak?
I want to thank Mr. Edo and Mr. Kathleen for today's lecture. My question is, if we were to apply this model in the environmental application, I think there's multiple variables that we need to include in their model. Like, for example, the influence of environmental stresses like pH, salinity, humidity, like heavy metals, like that Mr. Caroline said before, and I think that's my question for this.
Okay, that's a really interesting questions. In biology, we have like so many models. Like mathematical model is like so fast, like a very huge amount of mathematical models that's related to biology. So you can really choose like a model that's simple enough to explain or complex enough to predict. So you can choose. But if you want to consider like a stressors like a pH, a molarity, and also temperature, that's another problem because we don't have a law for that. So for the nutrient, you have this very nice relationship of how good the nutrient are and then how fast you can grow with that nutrient. But unfortunately, with the temperature and something like that, it goes like very random, and there's no like a law for that. So that's another thing. But it doesn't really affect like the differences between. It doesn't like significantly impacted this general law. So in the case of when you want to know, let's say you go to Kenjeran and you want to know like how fast the bacteria are growing in Kenjeran water, then you can take like some of the sample and then measure the Vibrio. You do some sorting. You get individual mass measured, and then based on knowing like the mass, for example, let's say like 60 femtogram, you can backtrace it to this law, and then you will get, okay, how fast they approximately grow in nature. So that's the idea. You can either choose like the model that's simple enough to explain or complex enough to predict. So you can choose. It depends on what you need.
Okay. Is there any other question from the audience? Oh yeah, silakan.
Oh, this is going to be interesting again. If cells are analogous to economic system, how do cells decide on resource allocation for specific metabolic processes when resources are limited, and do these decisions always results in the most efficient condition for growth? And then the second one, who created the genetic code? Why the OG pattern, etc.? How is it formed, and what is the relationship to bacteria and humans? Oh, wow, this is so philosophical. The second one is so philosophical, but let's start with the first one. If cells are analogous to economic system, how do cells decide on resource allocation for specific metabolic processes when resources are limited? So, there are so many ways, and one of the ways that people think that bacteria is sentient being that have a thought that can decide, okay, let's grow faster, or let's have an efficient way to allocate the resources. But that is not the case, because if you see that mechanistically, it's all just interactions of thing that is optimized by evolutions. So, and evolution is super random. They fit to what the fittest that survives depending on the environmental itself. So you cannot really choose, okay, which one is like the most efficient one. Sometimes you don't need to be efficient, but you need to be robust. For example, like in extreme environment, that is not the thing that they can decide to do that, but it's super random. It's what in their the genetic code within the cell itself. So when we make analogy, it's just a way to understand easier, like how they allocate resources. For example, let's say like Escherichia coli, you grow them on acetate, and then versus growing in glucose, and then why in glucose they grow faster. And this economic principle analogy can explain that better and make it easier to understand, because, oh, okay, I grow in glucose, they have like a shorter pathway to go, and then I have more investment that can goes into building more cells instead of like focusing on metabolism to get more energy. So that's why we use these economic principles, not the other way around. And then who created the genetic code? I don't know, man. I don't know either. it's from the evolutions, and then that's somehow that's the most efficient one, and that's somehow that's the thing that works. So evolution is not the thing that organism can decide. It's just like totally random. You have like more variations, and somehow only few groups that can like survive, and that's go selected and go like continue to make the offspring, and that's it. And I think the second one is very philosophical. And how is it formed, and what is the relationship to bacteria and human? Well, I don't know, man. I don't know either about that. But that's an interesting question, absolutely. Thank you very much. Dari siapa ini angkatan 2024? Okay. Any other questions?
Is there any other question from the students, audience? I have another question. So, for the bioreactor and also for Edu, for the bioreactor, do you design it only for aerobic condition, or we can use it for anaerobic things, or?
Thank you. Thank you. Thanks for the question.
We saw some bakso or some.
Yeah, it's in WhatsApp. It's a bit higher. You can show our secrets, I think. Yeah, a bit higher. No, I sent you. Yeah.
Sorry to make you open your
It's
WhatsApp
okay.
chat.
No, it's okay. We don't have. Yeah, you can see the transport here, the customs. We don't have any. Yeah, over there, lower. So yeah, this one. Yeah. So Edo's colleague is running anaerobic bacteria, anaerobic experiments, and they have an incubator in a tent, in an anaerobic tent, and the incubator is very narrow. And the device didn't quite fit, but this was fixed by printing a version where the pumps sit on top of the valves. So they're actually detachable, and it's not fixed like here. I can lift it with one hand and it holds together. And if Edo shows the next picture, you can see that the pumps from the top can also be put on the side, and this then fits very well inside the anaerobic tent for hypoxic experiments.
Nice to know that it can also be used for anaerobic things. Wow. Very well developed bioreactor.
Yeah, thank you. Yeah, it's a
Okay,
pleasure
yeah.
getting your interest.
It's very interesting, honestly, yes. Because I'm currently working with the gut microbiome things, and I'm wondering whether it's also possible to use this as a thing to simulate the condition in.
Yeah, so this was exactly for a group studying the gut microbiome.
Okay, nice to know. Okay, another question for Edo for this attaching or clumping cells. Did you do some pre-treatment or something like that to make sure or to reduce the number or to minimize the clumping cells, or you don't need to really do anything for that?
That's a tough question, Arif, because another consideration is that, for example, if you want to use like a detergent, for example, to like somehow loosen the cell that attach to one another, you might lose some of the mass inside the cell, and then lowering the mass, and it's not become accurate anymore for the mass measurement. So that's another trade-off that I have to do, like either choosing that a really single cell mass, but lowering the cell mass by itself, or having a clump cell, but more accurate, but I still don't know how to decide that. So for now, I just use the microscopy just for sanity check.
So to make sure the percentage of the population, which one is attached to each other.
Yeah, exactly. To see like approximately how many cell are clump, and then how many like groups, like for example, like duplets or triplets or something like that, to decide.
In your experience for Vibrio, how much percentage of the population is clumped to each other?
It really also depends on the species and also the nutrient they're growing. For example, like there's this a vegan Vibrio called Vibrio briogani. Yeah, the story. This is really interesting because this Vibrio cannot grow on amino acid. They cannot grow on amino acids like any other Vibrio, and so we call it a vegan Vibrio. Those are clumping really bad, like really bad. It's so nasty. I don't want to work with that. Another one that is really good that doesn't really clump is Vibrio cyclitrophicus, and also Vibrio, I forgot the other name, but those are really good as well. And when you grow that on glucose, they don't really attach to one another. So it's like a really planktonic, and then it's really nice, like a single cell. So it really depends on the nutrient and also the species itself.
Okay. So even vortexing is not really working pretty well, right?
It works, but it's just like reducing like a small amount of a clump cell. But still, it's kind of hard to work with.
Okay. Thank you very much. Is there any other question from the audience? Ada pertanyaan? Kalau mau tanya bahasa Indonesia juga boleh.
Kalau mau tahu, mau tanya tentang PhD life atau apapun yang mungkin nggak berkaitan sama ini juga nggak masalah. Feel free to ask any questions.
Iya, yang belakang dulu ya.
I'm sorry. So the module for the pump parts or data simulation yourself?
So the peristaltic pump head is a product that you can buy online, and inside is a stepping motor. And but that's a standard part. It's not something that I made.
Okay, so you just assembly, buy it online, and then assembly in.
Yeah, and I designed the device around like one specific model. But the valve is something that I designed myself. So something like this, you cannot
buy it in e-commerce. Okay, okay. Thank you. Other question? Jadi tanya?
I want to ask this. This lecture is about like economic principles of microbial growth, right? What if we go farther the line and we apply this this field of quantitative finance to microbial growth? Like, I want to ask, what's your view on this, please?
Oh, that's super interesting. Technically, economic and biology that somewhat overlap is that the mathematics that is used to explain the phenomena in biology. For example, like you can use like differential equations to see like how fast something changes. That also happened to economic, like how fast the economy is growing, and then you can somehow like predict, okay, after like a certain point, is it going to increase or decrease, or depending on certain situations. And also like in economy, there's this like Brownian motions like in stocks that can be used to like predict like the stock is going to increase or it's going to like go down, like what's happening in Indonesia right now. Sorry, but yeah, I'm not gonna go in political about into that, but yeah. So it's actually not applying biology to finance, but seeing that you can use math to either explain the biology or explain economics. So that's my view on this. So you can see that the math here is a tool to quantitatively explain the phenomenon, like for example, like the differential equation to see like how fast we're growing, and then what's happened in finance as well. So you cannot really like directly use what's in biology to finance or the vice versa, but some of it like have some analogy as well that you can use to explain better. For example, like resource allocations. That's a super overlap, right? Like, okay, are you gonna invest more on machinery or buying more cheap product? Or do you want to store more raw materials to produce cars? So that's the questions that somehow analogous to what's happened in the cells.
Thank you, thank you.
Yeah, thank you. Is there the question? Yeah.
Thank you for the opportunity. My name is Dave, and I'm really interested with that bioreactor machine, especially since that already explained earlier that the material, the reactor is using bacteria as the material. So my question is it possible that we can use the alternative material like yeast or microalgae for that machine? And then for the sterility protocol, I read that the machine use autoclave fluidics, and it will, and inside that machine there is a gamma ray that will kill all the microbacterial. Maybe can explain the detail a little bit more about the sterility protocol. Thank you.
You get the question right, Katalin?
So one question is about the sterilization protocol.
Yeah. And
right?
another one is whether your reactor can also be used for microalgae, yeast, or any other micro.
Yeah, so I've run many experiments with yeast already. With the algae, I haven't tried, but in principle, there's nothing that would stop you from growing anything. We're also thinking to start growing mammalian cells. And that's a little bit of a different kitchen because mammalian cells are very big and very sensitive. So you have to be careful with stirring, and it's very expensive to run the media for them because they don't just grow on cheap LB. So a contamination would be very expensive in mammalian cells. But with microalgae should be super easy. And the sterilization protocol, so I mentioned gamma ray during the presentation because that's just an interesting sterilization mechanism. We don't use gamma rays, we use just autoclaving. So do you have an autoclave here in general? So are you familiar with autoclaving or not really? No. Okay, so when you to autoclave something, you need to basically boil it in a pressure cooker, and you need to make sure that it's wet steam in contact with all the surfaces. And when you have wet steam at 121 degrees for 20 minutes, that's considered good enough to kill everything. And to achieve that inside the tubing, you have to make sure that there's at least a little bit of liquid in one of the vials that can then boil and evaporate, and the steam should touch all of the inside of the tubing. Other than that, you just you take the system, put it in the autoclave, make sure it's all of the pores are covered with filters. So here I have a 0.45 micrometer filter, and when you're starting the experiment, you connect this to a bottle. So you unscrew it, and then you connect it to a bottle aseptically, right? So this is the one movement that you have to do very carefully, and yeah, that's about it. You put it in the autoclave, wait for the cycle, take it out, carefully connect the connector, and that's it.
Okay, thank you.
The, yeah, maybe use the microphone so everybody can hear.
Okay, I want to ask one more thing. How can we know the stress management of the bacteria inside? That I know that by the bioreactor machine that has powered by Raspberry Pi,
Yeah.
and did that machine is informing us the notification like, oh, the tube files 1 is, or the bacteria already stressed, so we need to add drug or some fresh media, or it's like what something.
The device does the addition automatically. So you can program the regime in which you want it to increase the stress. You can say when it grows with this speed, it's considered healthy growth, and I want to increase the stress by 10% or 1%. And so when certain conditions are met, you say I add a certain fraction of stress, right? And when you do this multiple times, if you take like a number to, like if you take 1.1 to the power of 50, you get a very large number, right? Just by small increases. But yeah, in earlier versions of the device, I was doing this manually. So I would look on the dashboard at home and see, okay, this one is growing a little bit fast. I need to increase it. And then I got tired of doing it and I automated it. So it's now the device fully handles it. Yeah, I think it's yeah, maybe. Yeah, sure. Yeah, maybe like let's go through some more questions because I need 2 minutes to connect everything, and yeah, I'll start something to see it in action.
Yeah. So in some minutes we will see some demonstration of the bioreactor. While waiting, if you have any question, just raise your hand. Ada pertanyaan kah? Yang belakang sudah kepanasan ya. Mau tanya? Ya.
So this is a question about the machine itself. I was wondering why they were not just top of the why they were not at the top of the things like the small bit of indentation. I kind of wonder what it's for specifically. Yeah, like the indentation, like the notch. Why is like
Oh, that's the pins.
Can you? Could be. Think? Mm-hmm. 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 Can you let me? I want to share the screen.
Yang duduk belakang, are you okay? Nggak apa-apa? Makanya enak duduk depan ya. Udah dibilangin.
Okay, so a question arose about the notches here, right? If you, it's fine, it's fine. At the top, there are these notches where the silicon tubing goes. And the question was, why do we need these notches? Because inside is this valve. So if you look carefully at the video, when you, yeah, wait, it's showing over, yeah, but not. Okay, so if you look here, this is the tubing coming. Oh. Is it? Sorry. So this is the notch, and the silicon tubing goes in, and here you see a red little pincher. And this red piece of plastic presses against the silicon tubes to shut it off. Right, so this is how we control the flow of liquid to only one single vial. And because it's this kind of pinching, it's on the exterior of the tubes, so it's aseptic, right? You can do this even though the plastic itself is not sterile. The inside of the tubing is sterile. And when you press a button, it opens over there. Right? I'm gonna show that the second once again. Yeah, here you see it better when it just releases, and you can actually see the 3D printed lines. So these are the layers in the 3D printed part on this red bit. Yeah, I hope that answers the question about the pincher.
Okay. So the pincher is tight enough for the gas? Is it only for fluid things, or can we use it for the gas?
It's tight for both. And actually, the way to test it is with a syringe. So if you connect a syringe over here and compress it, the air is not going to go through. So it's you can compress up to about like 1 bar above atmospheric, and it holds. And when you open the valve, you can hear the sound of the air flowing.
Right. I have some technical questions. How much volume can you put in this glass? Maximum volume.
The total volume of the vial is 40 milliliters.
14.
But 40.
40.
40. But the working volume is anywhere between 12 and 30. And there is actually a trick. So if you can also have a very low dead volume. So now there is a long needle. The waste needle reaches about halfway through, and it leaves 12 milliliters below the needle. But for some applications, for example, for bacteriophage experiments, you need a low volume. And you can do this by a very long needle that reaches all the way to the bottom, and then you can really extract all of the liquid out of the vial. And for bacteriophage experiments, you need to do this because you want a dilution rate that is faster than the growth rate of bacteria. Because if you don't dilute them fast enough, you'll have phage-resistant bacteria that appear in the vial, and they take over the population. And you need to wash them off. And so the rate is faster than the growth rate of bacteria, but less fast than the growth rate of phages. But because phages rely on other cells and they can have a burst size of like up to 100-ish, they can actually replicate very quickly. Yeah, so volume is 40.
Thank you very much. Is there any other question from the audience? Both from Edo's talk or about this sophisticated reactor? I think, okay. Sudah ya. Okay, I think we reached to the end of the talk for today. So don't forget, tomorrow we will have another talk about the bioinformatic things. If you would like to join, please also maybe bring your laptop, right, Edo? Yeah. Ini nanti. Okay, yeah. So after this, please also fill the feedback form. It will be important for Mas Edo's report yeah, to the university. Last time, if you have any question, no? Okay, then I think we reached the end of this talk. Thank you very much for your time. And also, of course, thank you very much, Mas Edo and also Kathleen for coming here and your willingness to give talk to us.
Thank you so much for inviting.
Anybody, if you are interested to see, you can just come and then. Wow. It's interesting small playthings.
So now I started an experiment. This is what would be happening in a real experiment. You can see the stirrers are making a lot of noise because there's no liquid and they cannot hold the center. When there is liquid, they're more silent. And a pump is now pumping something. It's not doing anything because the tubing is not installed, but you can see the process. So it's every 60 seconds, you see the red lasers shine in all of them to measure the optical density. And also there's a lot of pumping in the process. Yeah, thank you so much.
You are very welcome. Oke. Oke ya, kita tutup acara hari ini. Terima kasih atas kehadirannya. Jangan lupa besok datang lagi kalau mau join. Saya tutup. Wassalamualaikum warahmatullahi wabarakatuh. Thank you very much. Ayo cepat-cepat ngisis. Oh iya, linknya di mana? Oh iya, link untuk feedback minta tolong diisi ya, sudah dikirim ke setiap angkatan. Terima kasih banyak. Oh ya, sebelum kalian cabut ya, mungkin berdiri kita foto dulu. Buat SPJ ya, dan juga buat sosmed. Kapan lagi kalian foto sama PhD student dari Austria ya?
Maybe Can you also press this button?
Kathleen, you can stand over there and then we can take a picture together. Yang lain berdiri apa duduk, Ira? Ya, yang belakang bisa berdiri. Berdiri, berdiri, yang belakang berdiri. Ya, minta tolong ya kerjasamanya. Ayo, ayo. Ayo harap ke kamera, hadap ke kamera, HP-nya ditaruh dulu ya. Thank you very much. You can
Ya, langsung deh.